Oh, welcome, and let me welcome you all to the Faculty of Health and Medical Sciences 3-Minute Thesis Competition Awards presentation. We are in a room here with very limited audience, but we are extending a warm welcome to all the audience joining us via the live stream today. My name is Rutger de Zoete, and I will be taking you through the proceedings today, and I'm very honored to do so. Before we start, I would like to acknowledge and pay my respects to the Ghana people, who are the traditional custodians of the ancestral lands we meet on today. We acknowledge the deep feelings of attachment and the relationship that the Ghana people have with their country, and we respect and value their past, present and ongoing connection to the land and cultural beliefs. So in preparation for today's session, we reviewed last year's awards ceremony, and my colleague Sean Martin was standing here that day and said, we were hoping to find ourselves in a unique situation, referring to the live audience and very limited audience in the room. And yet here we are, another year impacted by COVID, very limited audience and live stream. And indeed, we're all very much impacted by the situation, and we're struggling to get through this all together. We very much acknowledge that the candidates who have participated in this year's 3-minute presentation competition are experiencing those similar difficulties. Personally, for myself, not being able to see family interstate and overseas is a challenge, and we know that many of our international and indeed also domestic students are experiencing also those similar situations. Professionally, we've been moving from online teaching to face-to-face teaching, which has been an interesting situation. Research projects have been placed on hold, and international PhD students are struggling finding their place here in Australia. For myself, I have an international PhD student who is starting their PhD remotely, working through the first couple of months of their PhD remotely, not being able to be here. As you can imagine, that's a huge struggle, and we know that many of our students in the faculty have experienced similar struggles. But today is a moment of celebration of our research community and of the incredible work that is coming out of the faculty. I've been involved in the 3-minute thesis competition at 3 Australian universities in 3 different states. So I think I'm a little bit able to compare what is happening at different universities. And last year I joined the University of Adelaide for the first time, being part of the panel judging the presentations. And I was really pleasantly surprised with the quality of the presentations that were submitted in the faculty and across the university. I think and I know that supervisors and teams of the students here are very proud of what these students are doing. You might remember from last year's faculty winner, Kalia Primer, who went on to represent the university or the faculty in the university finals. She ended up winning the university finals across all 3 faculties. And I think that's a clear presentation of what talent we have in our faculty. And definitely we have seen the same quality of presentations this year. So we received this year 23 outstanding presentations submitted to the 3-minute thesis competition within the faculty. And today we're going to view our top 6 presentations and award 2 faculty winners. And those 2 faculty winners are going to proceed to the university finals in September. So for those of you who might not be familiar with the 3-minute thesis competition, this is really a competition or an exercise in research communication. And HDR candidates are asked to communicate their research and the impact or significance of their work to a non-specialist audience using a single static PowerPoint slide. So the competition was started in 2008 at the University of Queensland, and over the years it has really become a global event. Now the 3-minute thesis competition is held at over 900 universities across more than 85 countries. And as you are familiar, since last year we have temporarily moved this event online. It's a virtual format now, but the judging criteria are very much the same. Communication of those research findings. And those rules indeed have also stayed very much the same. So I think you all know by now the presentations are limited to 3 minutes, and there's a single static slide permitted to guide the presentation. No transitions, no animations, not anything moving on that slide at all. Presenters are not allowed to use any additional props, and the presentations can only be spoken words, so no poems, no songs, and unfortunately no raps. The decision of the panel, which we'll be discussing later, is final. So the panel has a really difficult task to differentiate between 23 outstanding presentations, and there are certain criteria that they use for that. So firstly, comprehension of the content. So did the presentation provide a clear background, and was the significance of the research project clearly articulated? Was the structure of the presentation clear and logically sequenced? And did the presenter spend enough time on each of the individual aspects throughout the presentation? The panel also judges on engagement and content. So did the presenter make the audience want to know more? Did the presenter show enthusiasm for their research project? And indeed also the PowerPoint slide was assessed. Was it clear? Was it concise? And did it really help the presenter to get across that message clearly? We'd like to note here that despite these presentations being recorded, the competitors will not be judged on the quality of the video recording or of their editing skills. Still, this is a competition for research communication, so it is really a focus on the presentation skills and the slide itself. So this was the panel with a very difficult task this year, judging these presentations, and we've got some of the panel here today, and we'd like to thank you very much for your contribution this year in assessing these presentations. And from first-hand experience last year, I know how incredibly difficult it is to differentiate between presentations that are also so well prepared and well presented. I think it is time that I stop talking and that we're giving the stage to the 6 top presentations of our faculty. We're going to start with our first presentation by Janine Tam from the School of Biomedicine. And the title of Janine's presentation is Silencing the Alarm of an Inflammation Invasion. We're first going to take a couple of moments to look at Janine's slide, and then I'm going to play the presentation.
Behind this wall is a battle against cancer. At the forefront, we have chemotherapy, trying its best to kill the cancer. But like all battles, there is collateral damage, and civilians are dying. In this case, the civilians are healthy cells that are unintentionally killed by chemotherapy. This causes the most common side effects of treatment, things like diarrhea and vomiting, which not only make people miserable while having treatment, but reduces their chances of being cured, as chemotherapy often has to be stopped or reduced. But how exactly does this happen? Of all the tissues in the body, the gut has many civilians susceptible to being killed by chemotherapy. But the gut is savvy and has employed its own army to help defend itself, led by none other than TLR4 that sits in the watchtower. Here, TLR4 watches for danger signals from chemotherapy and other invaders like bacteria, and sounds the alarm for reinforcement against the damage. It's this alarm that calls to action the body's immune system army to eradicate these invaders and heal the collateral damage from chemotherapy. But TLR4 has a tendency to overreact and kills the invaders at the expense of maintaining gut health. So put simply, in trying to protect ourselves from the damaging effects of chemotherapy, this army actually exacerbates the gut injury. My project is focused on controlling this collateral damage by dampening TLR4's over-the-top alarm response. And how am I doing this? Well, I am recruiting the lollipop man over here. He is an inhibitor sent to silence the TLR4 alarm. With this gone, there won't be any reinforcement from the immune system army, which would significantly reduce the amount of healthy gut damage. Then chemotherapy would kill the tumor, without causing collateral damage, which would reduce the side effects of chemotherapy and ultimately improve the quality of life of people with cancer during treatment. Thank you.
COVID-19.
Thank you, Janine. That is a really wonderful start. All right, our next presenter is Tassia Oswald from the School of Public Health. And the title of her presentation is Green Views for Screen Blues. We'll have a look at her slide first. And then we'll start the presentation.
COVID-19 has changed life as we know it, and mental health consequences may be one of the greatest impacts of the pandemic. However, research tells us that depression and anxiety were already on the rise among young people before the pandemic began. Experiences of mental illness in adolescents are linked with poor mental health across the lifespan. So, we really need to prevent mental illness before it develops in young people, rather than just treat it once it presents. So, what has caused this rise in youth mental health problems, and how can we help prevent it? Screen time is a unique feature of contemporary adolescence, which is regularly argued to be linked to this increase in mental illness. Young people report spending an average of 7 hours per day in front of a screen. And alongside these increases in screen time, young people's time spent in nature, or green time, has also decreased. Young people report an average of 6 hours of weekly green time, which is less than their daily screen time. But what evidence is there that these trends actually impact on mental health? Well, in my PhD, we brought together evidence from almost 200 international studies. We found that according to majority of the available evidence, high levels of screen time are linked with poorer mental health, while green time is linked with better mental health. But why might this be? Well, from an evolutionary perspective, we evolved to function best in natural environments over millions of years. But this extensive evolutionary tuning is lacking for tech environments. When in nature, our brains use bottom-up processing. This means that we're using the most ancient parts of our brain at the bottom. This is quite relaxing. It can boost our mood, restore our attention. But on the other hand, during screen time, our brains use top-down processing. This means we're using newer parts of our brain around the top. And this is quite demanding. It can fatigue our attention and make it harder for us to manage our emotions. Given screen time and green time affect mental health in such contrasting ways, I asked, could we use green time to help buffer the psychological consequences of screen time? And that's what the final phase of my PhD is looking at. We've been getting adolescents to complete measures of mood and attention, first at baseline, second after a period of screen time, and finally after a period of rest, either in an indoor setting or outside in the botanic gardens. Data collection is ongoing, but we anticipate that adolescent mood and attention will decrease after screen time due to that demanding top-down brain processing. But after the rest period, they'll be restored. However, we hypothesize that attention and mood restoration will be greatest for those who are outside in the botanic gardens due to that relaxing bottom-up brain processing when in nature. If our hypotheses turn out to be true, nature immersion could be a great way for young people to support their mental health in an unavoidably high-tech era. Green views could be the perfect antidote to screen blues.
What would life be like?
Thank you, Tassia. That's a wonderful presentation as well.
COVID-19.
So we're moving on to our next presenter, Sonia Nath from the Adelaide Dental School, with her presentation titled Microbiome Recycling. This is Sonia's slide.
What would life be like if you and your kids had no tooth decay? No need for painful visits to the dentist, and we would all love to say bye-bye to dental bills. It's early morning, we wake up, still in our sleepy pajamas. What's the first thing we pick up? It's our humble toothbrush, cleaning the sticky stuff off our teeth. What exactly are we doing? We're removing all the excess bugs that collect on our teeth, the dental plaque. Some of these bugs are responsible for tooth decay. Can we really get rid of them? Well, not really. Microbes are present everywhere, on our skin, hair, gut, and especially teeth. Over millions of years ago, a partnership was formed between us and the microbes. It's like a contract to protect each other. So are we still honoring the ancient contract? Certainly not. We use antibiotics and chemicals which can affect the delicate balance with our microbe friends. Imagine your mouth as a primitive rainforest. Leave it at peace, the nature restores itself. When we humans intervene, cause a havoc, destroy everything, the good, the bad, leaving no diversity. So what can be done to avoid the effects of the nasty chemicals and overrun the bad bugs which are causing tooth decay? Simple. We bring in the army of the good bugs. What if I tell you today the bad bugs in your mouth can be changed into the good bugs by simple transplant? And this is the important research I'm working on. My research is called as OMT, Oral Microbiome Transplant. Microbiome is a unique mix of microbes and the genetic material. When present in the mouth, they're called as oral microbiome. We're going to transplant good bacteria from a healthy mouth to a person with bad bacteria that are causing disease. I'll start by identifying the superheroes, people who have the great oral microbiome. I will extract the dental plaque, clean it, and mass produce it in the lab. My research will make sure that this is safe, effective, and will determine that it actually works. I will start my research with the lab animals and then finally humans. So far, preliminary results have shown the successful growth of the healthy bacteria, and it's an important first finding. Oral microbiome transplant may be the game changer that can give you a healthy mouth and a lifelong protection from oral diseases. So next time you check your teeth, remember you could hoard a wealth of health.
Diabetes.
That's a great presentation. Thank you very much.
Would life be?
So our next presenter is Sanuri Liyanage from the Adelaide Medical School. And she's presenting on finding a new savior in the war against diabetic foot amputations. She has submitted this slide. And we're going to have a look at her presentation.
Diabetes is the silent pandemic of the 21st century. Around the world, more than 1 million diabetes-related limb amputations occur every year. So why are diabetic patients at such a high risk? Diabetic wounds or foot ulcers are notoriously hard to heal due to insufficient blood supply. The inability to form new blood vessels to supply oxygen and nutrients makes such a toxic environment for any cell to survive, leading to tissue death and amputations. Currently, there are no effective therapies to prevent this devastating outcome. But my PhD focuses on a local hero that just might help do exactly that. I have found that a fundamental reason why diabetic wounds have such a hard time growing new blood vessels is the gradual decrease of a local stem cell population. Think of a stem cell as your body's blueprint from which new cell types are formed from. What's unique about the stem cells I'm studying is that they've been living in your skin independently long before you were even born. And what's novel about these stem cells, or what I'd like to call their superpower, is its remarkable ability to transform itself into not just one, but two different cell types, vital for wound healing. A builder cell that builds new blood vessels, and an immune cell that supports the builder in their construction and also help fight infections, together leading to better wound healing. However, when exposed to high sugar levels, like in diabetes, our local hero begins to lose its superpowers. And a deep dive into why revealed that a malfunction in their mitochondria, the cell's powerhouse, just might be the reason. Though these stem cells are your everyday local heroes, sometimes all heroes need just a little bit of help. I'm working on delivering a targeted therapy to help rescue these stem cells in diabetes by restoring their powerhouse or by transplanting healthy stem cells into diabetic wounds to restore their construction ability and lead to faster, better wound healing. What makes them an excellent candidate for stem cell transplant therapy is that there is very little to no risk of your body rejecting them. I believe this takes us one step closer to win the war against diabetic amputations.
Thank you very much. That's a wonderful contribution. So next presenter from the School of Biomedicine is Darren Chow, and he's presenting on embryos with a light touch. Have a look at the slide. And we'll start the presentation.
Let me tell you a story about Luke and Amy. They are really eager to start a family. However, after many attempts at conceiving naturally, they were diagnosed with infertility. Infertility affects more than 15% of Australian couples. It is often an invisible and stigmatized health challenge. This means that you or someone you know may be suffering from infertility, be it a family member, a friend, or a colleague. Luke and Amy went through multiple costly and emotionally draining IVF treatment, which weren't successful. Strikingly, only 1 in 5 initiated IVF cycles will result in a baby, and the success rate has remained stagnant for several decades. A key factor for IVF success is a woman's age at the time of treatment, as infertility increases with age. What may also come as a surprise is the incidence of aneuploidy, or plainly, cells not having the expected number of chromosomes, also increases with age. In the IVF clinic, the current gold standard to detect aneuploidy involves taking a small number of cells from the embryo, also known as a biopsy, and then sequencing the DNA to confirm whether the embryo has the predicted number of chromosomes. Apart from being highly invasive, this procedure is also notoriously inaccurate and is also known to predispose the mother and the baby to a lifelong increased risk of cardiovascular disease. My project aims to address this shortcoming and explore an original notion. Can we use natural light scattering from the embryo to identify aneuploidy non-invasively? Light scattering is all around us, from the blue sky to light colors reflected from a puddle. This scattering gives rich information about the particles in the sky or the water in the puddle. Interestingly, healthy and sick cells have different metabolic property, which is reflected in the way they scatter light. I have developed a new way to image that involves shining gentle doses of light upon an embryo and using the scattered light that comes back to reveal the biochemistry of each cell at high resolution. Meaning, we can see minute details, allowing for the identification of healthy and sick cells in an embryo, all in a gentle manner, a light touch. This technology enables us to locate aneuploid cells and even work out how many are present within an embryo. This research will change the way that aneuploidy is currently being diagnosed in the clinic and also help clinicians to choose the best embryo that will give the best outcome, which is the birth of a healthy baby. Collectively, this project is well positioned to improve IVF success and consequently the emotional well-being and accompanying societal benefit for couples like Luke and Amy. Now, before I go, allow me to leave you with this question. Would you like to know the health of your embryo?
Great presentation, Darren. Thank you very much. Our next and final presenter is Thomas Litster from the Adelaide Medical School. And Thomas will be presenting on intellectual disability, putting the pieces together. Have a look at his slide. And we'll watch Thomas's presentation.
The human genome is one of the most complex puzzles that we've ever tried to solve, but for many families it's much more than that. For those affected by genetic disorders such as intellectual disability, solving the puzzle that is their genome can lead to the identification of a disease-causing mutation that is so heavily affecting their lives. To identify these disease-causing mutations, we need to try and reconstruct the genome of an affected individual. and that's where the problems start. Typically, this reconstruction is done using short fragments of DNA or short read data. This is like trying to solve an enormous puzzle with tiny puzzle pieces. It's not going to be very effective. Look at how it's worked here. Using the small puzzle pieces, big chunks of the picture are missing, important pieces of this puzzle haven't been placed, and the small size of the pieces obscure a lot of really important information about this family, and it's much the same when it comes to genetic analysis. Using short read data, we're probably missing big chunks of the genome, which could likely house the disease-causing mutations. Fortunately, advances in technology have allowed us to sequence much longer fragments of DNA. This allows us to generate larger pieces to try and solve these puzzles, and that's where my research comes in. I'm working with individuals that have intellectual disability, but haven't had a disease-causing mutation identified yet. I'm taking their DNA and sequencing it using Oxford Nanopore's long-read sequencing technology to generate long-read data. Using this data, I've been able to identify mutations in genes associated with intellectual disability that haven't been able to be identified doing a similar analysis using short-read data. And what I'm hoping to do is further interrogate these newly identified mutations to identify the disease-causing mutations in the families that I'm working with and report it back to them. And you may be asking, why is it important that we know the specific mutation causing the disease if we already know that it's intellectual disability? And this is for a number of reasons. The first is that knowing the specific mutation allows genetic testing to be done on other family members, allowing them to make more informed decisions when it comes to future pregnancies and family planning. Another reason is that different types of intellectual disability are caused by mutations in different genes. By knowing the specific gene that is affected in the particular case of intellectual disability, it allows for the families to reach out to other families who might have gone through similar things to increase their support network. It also allows for doctors to make more informed decisions when it comes to treatments, and potentially opening the door to clinical trials which could improve the quality of life of these affected individuals. And so I ask the question to you. With so much on the line, if you're trying to solve these puzzles and make these diagnoses, would you prefer to use the small puzzle pieces or the large ones, which make it much easier to put the pieces together?
Great work, Thomas. Thank you. Great presentation also. I'd like to ask you to join me in thanking all the 6 finalists for the faculty today who have shown some wonderful work and great presentations. Thank you very much. We'd also like to extend our gratitude to the judges who have spent quite some time on working through these presentations and assessing the presentations, and thank you very much for your contribution in that. And we'd like to thank all the students who have submitted their presentations for this 3-minute thesis competition. Very well done. Everyone has done a very good job. We'd also like to thank the audience today here in the room, but also we'd like to thank the live audience who has joined us today via the live stream. Thank you very much. And in particular, we would like to extend our sincere appreciation to the Northern Communities Health Foundation for their generous support of this event over the years. I would like to invite Professor Gregory Crawford to the stage, who is going to come and present the awards to our 2 faculty winners. I think you will.
Thank you. I'd like to again acknowledge the Kaurna people as well, and thank you for inviting the Northern Communities Health Foundation to be part of the 3-minute thesis. My fellow board member, Professor Suren Krishnan, who's an ear, nose and throat specialist, is present. The Northern Community Health Foundation has a long and strong relationship with the University of Adelaide, and we're a board of about 8 members, administrators, finance, legal and medical and marketing expertise, and we have been trusted with a fund from the Northern Community Hospital when it closed and support various projects, not only the 3-minute thesis but the poster prizes for the Florey Conference, We established the Derek Fruin Early Career Research Award and have a visiting professor pre-COVID from usually overseas. This year we're having a large general practitioner update to do something locally. We provide funds for small research grants and a significant number of community capacity building projects. And we've traditionally had an interest in palliative care and mental health, and our focus for the coming years is going to be ageing, we've decided. So we're very pleased to be here, and I think Surin is going to actually give the prize, and will you announce them? Thank you very much.
Thank you very much, Professor Crawford. I've got the envelope here. This is the Academy Awards moment. I'm going to open the envelope, and two gentlemen here are going to hand out the awards. So we've got two winners, and those two winners are Tassia Oswald and Janine Tam. Congratulations. Come forward. Come over here.
Congratulations.
Thank you very much.
Swap
Thank
bags.
you so much. Swap bags?
Swap bags.
Thank you very much.
Congratulations. Well done.
Please join me in congratulating these two fantastic presenters. So that concludes today's session. Congratulations to our two winners, and congratulations to all presenters and competitors in our 3-minute thesis competition. You've all done a really good job. Thank you very much for joining us today here and via the live audience. And we would like to invite you to come and support our two winners in the University of Adelaide virtual 3-minute thesis final, which will be held on Tuesday, 14th of September. So please all come along and support our two winners there. Thank you very much for coming today.